Hello. How are you guys doing? Oh, there's a tiny part of me that's very nervous that when I click the go live button, nothing's going to happen. But it looks like something has happened. How are we all doing? Did I see that someone's from India here? That is pretty cool. I think that's the furthest away I've ever had anyone watching me do anything in my life. So congrats. Hey, Sabiashi. Hey, Nazmoon. Sorry, I totally said your name wrong. Hey, Bethany. Hey, Joe. Hey, Blair. Hey. Hey. How you doing? How have your days been recently? School been all right? Bethany, I apologize that this is starting just a couple of minutes after you got home from school. If I'm totally honest, I got home from work about an hour ago, so equally, I'm sort of putting everything together very quickly. Can you guys go on TikTok and give us quick facts and questions? I'm sure that is something that we are probably looking at the moment, since we've got Instagram and stuff. Looking for much more season in the near future. Lovely. Smash your phone today. Good job, Joe Daniels. Yeah, I feel like that is a very good way of summarizing how this week is going to carry on going. Hopefully it's not going to get any worse. Thank you, Cara, for asking how I'm doing. I'm doing really, really well, yeah. What have I got to say? I don't think I've got anything remotely interesting to say. Got a nice cup of coffee next to me, which has my face on it. When I left my old job, my colleagues thought it'd be funny to make a mug which has my face on it. But there I am. There I am. That's me as a corn on the cob, in case anyone wanted to see that. Cumbria, nice. Cumbria's pretty far north. Passed my class because Snap provides videos. What can I say? Anyone else got anything else to say? You had a biology test on DNA today. Nice. Oh, cool. Right, yeah, let's do this. So when are we uploading this? I'm pretty sure, Chelsea, this just sort of uploads itself. I'm pretty sure when I'm done, it just sort of goes up. Like I would try and say I know how it works, but I have no idea. Awesome. Right, let's have a look at some nerve impulses and synapses then. So something that lots of people find quite difficult from what I've sort of gathered over the last 4 years of teaching. So by the end of today then, if you don't do this, it's fine. But I'd like for everyone here to be able to tell me what I mean by arrest and potential, specifically how it is maintained, because that is the standard question. I want you to know what I mean by depolarization, and I want you to know how a impulse is propagated basically along a neuron. Okay, so I want you guys to know sort of some stuff about that. Can I cover photosynthesis and respiration? I literally did 2 weeks ago and the week before that, I believe. So have a look on YouTube and you'll see me covering it. I'm not going to do it instantly. Otherwise, I'm going to spend my life just doing photosynthesis and respiration. Okay, finally, we're going to describe how synapses work as well, because people tend to get a bit confused with this, just because there's lots of things that sort of follow after each other in a bit of a sequence. So for those of you that care, here are the spec points. I'm not really going to go through it. They're pretty much the same for all different specs. Feel free to come back at some point and have a look and just work your way through being like, oh, do I know the structure of a motor neuron? Yes. Do I know this? Yes. Do I know that? Yes. Do I know this? No. Maybe you spend some time on it then. Here's OCR. There's LXLA. There is LXLB. Again, very, very similar. The only thing that's a bit different is some of the specs don't cover inhibitory postsynaptic potentials, but I'm going to anyway, so it doesn't really matter. Is this covering the eye? I'm not going to cover the eye, no. Maybe in the future I could do something about eyes, because that is quite a hard topic. So when we next do like an Instagram poll, if lots of you want me to do the eye, then just make sure you write that you want to do the eye and I'll try and do it. Okay, cool. So resting and action potentials then. So I think this gets overcomplicated really quickly because everyone starts to rush in thinking about voltage and millivolts and all that kind of thing. But I think put really basically, just from a very start, it's important that you know that a membrane can either be at rest, so it can either be doing pretty much nothing-ish, so it's at rest, or it is undergoing an action potential. Okay, they are the two options. So it's either at rest or there is an action potential going. There is somewhere in between, which is called a generator potential, and I'll talk about that in a second. But when it is at rest, when the membrane of the neuron is at rest, and you guys should hopefully know that your motor neurons look something like this, where you got your cell body, and you got your myelin or your Schwann cells, whatever you call them, and you got your little axon terminals. In terms of the membrane of it, you guys need to know that when it's at rest, it is negatively charged. And when I'm saying it's negatively charged, because I'm talking about a voltage, and I'm sure some of you do physics, you know that a voltage is a potential difference. It's a difference between two places. So it is negatively charged compared to the outside. Can anyone tell me how the membrane manages to maintain this negative charge? So how can it be negatively charged most of the time? And it is -70. Yes, you are correct. Although, Lucy, some different animals can be slightly different, but lovely. Lucy, you are right. Olivia, you are right. So this thing here is the sodium-potassium pump. What does it do? What is this sodium-potassium pump doing? So it moves positive ions out. Outside positive, inside negative, yes. What about sodium and potassium? What's it doing? So sodium leaves through the leaky channels. Okay, cool. So there are some channels that potassium can move through too. Good, Emily, you've said what I want you to say. So in order for it to be negatively charged, all I need to do is have some means of pumping 3 sodium ions out for every 2 potassium ions that come in. And this is what this pump does. So it moves some potassium in and it pumps 3 sodium ions out. So let's get this right as well. So lots of people get a bit, I don't know, they lose marks here because they don't call this protein by its proper name. So it's the sodium ion / potassium ion pump, right, to give it its full name. And we can say that it pumps 3 sodium ions out and 2 potassium ions in. Okay, this is not facilitated diffusion. This is active transport. If you ever see this word pump always means active transport. So we could say that this uses the energy from the hydrolysis of ATP. Okay, so ATP is required to do this. Someone also said that there is this channel. So there is a potassium ion channel here. And this has the ability to basically allow potassium ions out. So you guys all hopefully know that anything that is charged cannot move through the membrane unless it moves through either a channel or a carrier. And this channel is here to allow potassium to leak out. Okay, so K ions move out, or let's change that, let's say diffuse out. Let's be little scientists about this. Down concentration gradient. Okay, so all of this means that lots of positive charge is going to be going outside. So here are these little particles which are moving out, these little sodium particles. So all the positive charge is moving out, and then a little bit of positive charge that does manage to get in, the potassium, is just going to leave anyway. So all of this means that the inside is negative compared to the inside. Right, let's look at some comments then. So isn't it necessarily negative? Is it just less positive? Yeah, so with this, because I'm talking about voltage is a potential difference, right? And I know lots of you are saying, oh, my teacher said this, my teacher said that. Your teachers are right to say it's less positive or less negative or whatever, because that's what a voltage is. It's just a difference, right? So to say that the whole thing is negatively charged or positively charged, that might be an oversimplification. What we're saying is that the inside is more negative than the outside. There is a potential difference, hence there is a voltage. Okay, and it is -70. Let's write -70 somewhere. Millivolts. Okay, so ultimately, yeah, this is what you guys were kind of just saying to me. You end up with this situation where the outside is more positive than the inside is. Okay, so we can say, therefore, that the membrane is polarized. Yeah, because there is a charge difference on either side. And now, hang on. I've noticed that I do this thing where if I start talking whilst I'm writing, I end up writing the thing that I'm saying. So I probably should stop doing that. Yeah, so you can see, look, there are some positive charges inside, but on the whole, most of the positivity is outside. That's because mostly I've got sodium coming out and I've got a little bit of potassium coming in, but the potassium can move out because the membrane is permeable to potassium. Muy bien. Okay, so if we had an action potential, then what are the sequence of events that are going to happen? So my membrane tends to be at rest if nothing's going on. If suddenly there is an action potential, what happens? So Lucy, spot on, you are right. Good. So first thing that happens then, let's imagine someone mentioned eyes earlier. Let's imagine that somewhere down here, in fact, eyes are actually a terrible example of this, and those of you that have studied eyes know why. Let's say this is like a Pacinian corpuscle, which is essentially this weird sort of spirally thing which has a neuron in the middle, and it's basically how you detect pressure, for those of you that haven't heard it. So if someone pokes you, it's something called a Pacinian corpuscle that responds. So let's say that firstly, a stimulus arrives at the neuron. So a stimulus is going to arrive. Okay, what happens next? So what have you said? Stimulus, sodium channels open, lovely. Sodium diffuses in. Influx of sodium causes it to depolarize. Depolarization, repolarization. Voltage-gated sodium ion channels. I'm intrigued at the use of voltage-gated sodium ion channels. I'll talk about them in a second. But lovely. So first thing that happens then, the stimulus arrives, and this causes sodium ion channels to open. Right, key tip here, and if I could give you any tips for this topic, this is one of them. Just get in the habit of writing Na+, right? So I've seen a exam from about 3 years ago where in the mark scheme, it said reject the word sodium because loads of people were saying sodium and not sodium ions. So just get in the habit of saying Na+, okay? So Na+ channels open. If that happens, we can say that the membrane is permeable to sodium ions. Okay, so the sodium channels open. As you guys know, because I've been pumping a whole load of sodium out for the last 10 minutes or however long. As you guys know, there will now be this concentration gradient, which has been established, and sodium can now diffuse through this channel. So this sodium is going to start to diffuse through this channel down its concentration gradient. Okay, so I like the word that someone said earlier. So influx of sodium ions diffusing in. I thought that was worded pretty well. Okay, if that happens, you guys know that this word, if it starts in, so let's say it starts off the inside being -70, if suddenly there's a whole load of positivity coming in, then this charge is going to start to get closer to 0, right? So let's say that it's going to get to roughly -55, and you guys know why I'm using that number. Let's say it's starting to get closer to 0. We can call that depolarization. Okay, so this process is called depolarization. Okay, so polarization being there is this difference in charge. Depolarization is that difference in charge is starting to get smaller. So it's starting to depolarize. And as lots of you have just started saying, it reaches something called a threshold. And also someone who said that this is facilitated diffusion, you are spot on as well. But it tries to reach something called the threshold. Now, if the depolarization gets to, let's say, -60. So let's say it only gets to -60. So it goes from -70 to -60. If it only gets to that number, then that is not enough of a difference. There's not enough of a depolarization to cause an action potential to be formed, right? It's not enough. If it gets to -55, if enough sodium moves in that it gets to -55, this is called the threshold level. Okay? And essentially what it means is if it doesn't get to that level, nothing will happen. So if you were to have your hand like out like that, and the tiniest little like insect in the universe landed on your finger, if it didn't like put enough pressure on you, then you basically wouldn't be able to get this threshold level met, and you wouldn't have an action potential, so you wouldn't feel it. Okay, so depolarization, if it reaches -55, and this varies a little bit from spec to spec, so be careful if this is the right number. But if it reaches -55, the threshold is met. Okay, and you might hear this given a term. Does anyone know the term that is used to describe this threshold? Like if it reaches it, it's called the something, something, something. -40 FedEx LB. There you go. There are always some differences here. Not the refractory period. What's this called? If you reach a threshold, you get an action potential. If you don't, it remains as a generator potential. Cool. This is called the all or nothing response. Okay, you either have everything or you have nothing, right? It's not summation either. It's not absolute refractory period. It's the all or nothing response. If you don't meet this all or nothing, if you don't, get to this threshold, then instead of making an action potential, you actually make something called a generator potential. So a generator potential is basically an action potential which hasn't quite met the threshold yet. Some of you will use that in your spec, some of you won't. But finally, if I do reach the threshold, what happens next? So if it gets to the threshold of minus 55, what happens? If it gets to minus 55, repolarization won't happen just yet. An action potential will happen, yeah. Repolarization happens. Something really, really important happens after this. The voltage becomes positive. No, at minus 55, it's still minus 55. Jasmine knows what she's on about, though. It causes more sodium channels to open further along the axon. Not necessarily further along, but it causes more sodium ions channels to open, right? And Emily, you are spot on that these are the voltage gated ones. So if it gets to this specific voltage of minus 55 millivolts, then voltage gated sodium ion channels open. Okay, so this first little channel here, this guy over here, which I don't know why I just scribbled it in, but that one, this isn't a voltage gated one. This is just a normal channel which can open or close. Let's say further along, let's say over here, there is another one of these, and this one can only open when it gets to minus 55. Okay, when it gets to minus 55, it opens, and they are the voltage gated sodium ion channels. Okay, someone mentioned earlier that this is therefore an example of positive feedback, one of the few that exists, because it's normally really disastrous, right? So if you get a little bit of sodium in, this is positive feedback because it causes more sodium to get in. Okay. What is the order of active transport facilitated diffusion? What do you mean? What is the order? I don't quite understand. And we'll talk about localized currents in a sec. Minus 55 for AQA, I believe. Yeah, but I could be wrong. Okay, cool. So following this depolarization, my cell has now got to a stage where, I don't know, it's no longer at minus 55. Let's say that it's got to roughly positive 30. So there's now so much sodium inside that it's got to positive 30. What happens next? Positive feedback is where you basically have, I don't know, how can I describe it? Say like with your blood sugar, if you end up having too much sugar in your blood, your body will try to get rid of that sugar. If it was positive feedback, you'd have a bit of sugar in your blood and your body would then make more sugar. So repolarization is going to happen next. Lovely. How does repolarization happen? Blair knows what he's on about. Good to see Blair. I like to say that Blair is someone who comes to a lot of my web classes, so he knows. Yeah, at a certain voltage, so at plus 30 millivolts, we can say that voltage gated, and I'm going to be lazy and use VW from now on. So if anyone asks me what VW stands for, I will murder them. So at plus 30 millivolts, voltage gated sodium ion channels close. Okay, so at positive 30, the voltage gated sodium sodium ion channels close. Okay, this means that my membrane is very much impermeable to sodium. Okay, so that we can say that the membrane is impermeable to sodium ions. What happens next? Sorry, so I'm sure lots of you said this. I'll tell you when refractory periods are coming up. So good. Voltage gated sodium ion channels close. Yes, I will murder you. I did say that. So potassium channels now open. Now, because it's got to a specific voltage, the potassium channels that open are actually voltage gated too. So we can say that voltage gated potassium channels potassium ion channels open. Now, the pump earlier was basically putting some potassium in, but lots of it was going out again. So if I start to open my potassium channels, what's going to happen to potassium? So there's lots of potassium which is being moved in, don't forget. Good. So potassium ions diffuse out using facilitated diffusion. So potassium's going to diffuse out, and we call this process repolarization. Yeah, so polarization, it's got a charge between the two different sides. It is repolarizing it. It's getting that difference again. Now, I'm sure lots of you know this. What's the cool, not cool, what's the like unique little fact about these potassium channels? And it is an electrochemical gradient too. So what's the interesting little fact about these potassium channels, which I've got over here? Good. So they take a very, very long time to close. So you might wonder what this little ball thing here is. This is literally what they look like. They have a little ball on a little chain, and basically this thing has to go and plug this hole to stop any more potassium from moving out. So they close very slowly, which means that my membrane not only repolarizes, but it will actually hyperpolarize. Okay, so that's because the potassium ion channel is very slow to close. So hyperpolarize, and this is where we have this phrase that comes up called a refractory period. And there are actually two types of refractory period, and only one spec cares about this. Most of the ones that you guys will know of is called the relative one. One is called the absolute. But essentially, because it's slow to close, you can't get a second action potential being made straight away. Right? It can't happen straight away because this is so slow to close and you've got all this potassium leaving. So the refractory period is where you can't get another action potential immediately after. Okay, so you guys are probably used to seeing something that looks a bit like this then. So I normally have my resting potential, which is like, I don't know, somewhere down here. So this is the rest, nothing's going on. My sodium potassium pump is moving sodium out, potassium in. Suddenly I'll get a stimulus, and look, here are some failed ones. So here are some little stimuli where a little insect landed on you, but it didn't actually manage to make an action potential. So these remained as generator potentials, right? All or nothing response, they ended up being nothing. So suddenly, if I meet this threshold of minus 55, then my voltage gated sodium ion channels open. It causes my membrane to depolarize. After it gets to a certain voltage of roughly plus 40, those sodium ion channels close and potassium channels open. This causes it to then repolarize. And because it's so slow to close, it actually goes a little bit below the rest, which is often called the refractory period. For those of you that do EdXL-A, and you'll realize why I'm telling you this in a second, I'm going to show you a question which kind of links to this. Technically, this whole period is called a refractory period, right? This whole period can be called a refractory period or the absolute refractory period, because during this time, the same bit of membrane can't actually have an action potential happening. Because if suddenly my sodium channels are open, I can't have another one immediately after because it's still doing this same action potential. So this whole thing is actually a refractory period. But for 3 out of 4 of the main specs, it's just this bit here, which is the refractory period. Okay, what are we talking about? Someone's never seen that graph before. That is shocking. So what are the two types? There's something called the refractory and the absolute. I'm pretty sure it's just EdXL-B that worries about this thing. Okay, so this is the relative one down here, which is one that most specs care about. And this one, the whole thing is sometimes called the absolute refractory period. I was going to write some stuff on this page, but to be honest, I don't think it really needs all that much. You just need to be able to look at this graph and sort of understand what it's telling you. Okay, and then the final thing about action potentials, which is really fundamentally important, is now we've said what depolarization is. So depolarization has happened here. How is it the case that my action potential can start off over A over here and end up going to B? Can anyone tell me this? Good. So these are localized currents. What is happening? So why is it that my action potential can start at A and end up getting down to B? Nothing to do with nodes around VA here. Nothing to do with saltatory conduction necessarily. So let's imagine that these neurons don't have myelin, to just make it nice and simple. Let's assume they don't have it. Good. Yeah, Emily, you're right. Someone else said it earlier, I believe. I can't remember who said it. But some of this sodium that moves in. So when we say that depolarization happens, everyone is very quick to say that depolarization equals sodium ions moving in, right? But that is not really the end of the story. Some of these sodium ions, they are going to diffuse along this way. So let's say that a few little bits of sodium are going to diffuse this way. That means that the membranes here are now suddenly going to meet their threshold. If they've met their threshold, then even more sodium is going to move in. Let's say that this sodium has moved in and it's now going to diffuse this way, right? It means that this bit has now met its threshold, so more sodium can move in, right? And you have this constant wave or this propagation of action potentials because sodium is constantly being able to move in because it's diffusing. So it's really important that you guys know that the way that nervous transmission works is because a little bit of sodium is going to diffuse sort of laterally, or it's going to diffuse horizontally or to the right or whatever along the neuron. Okay, so some sodium or some sodium ions diffuse to the side, causing the threshold to be reached. Oops. Okay. Can anyone tell me why it can't go backwards? Ghosted Guna, this is convenience. So yeah, I have my wave depolarization, some Na diffusing. This causes the threshold to be met, causing action potentials and a wave or a local current. Why can't it go backwards though? Good, because of a refractory period, exactly. So because my sodium, let's talk about this image down here, right? Because that's the one I haven't scribbled all over. Let's say that some of my sodium here, right? Let's say that some of it tries to diffuse this way, because it will, because diffusion just happens randomly because of Brownian motion, right? So some of my sodium is going to move this way. However, because this bit is currently in hyperpolarization or repolarization, it means that it can't actually have an action potential again. So if I'm in this state over here, where it's gone massively below the resting potential, so it's like even further below minus 65, then let's say that normally you only need that much sodium to make, hopefully you guys can see me, like I'm up here. If you only need that much sodium to cause an action potential, if you're already like 40 millivolts below the resting potential, then instead of that much sodium, you're actually going to need this much sodium. And obviously this is a random amount of this unit I'm showing you. It's not a real unit, but you get the point, hopefully, that because it's already below the resting, it means that you'd need such a big like receptor to have such a big response, such a big stimulus, that it's just never going to happen, right? So it can't go backwards because part of the neuron is in the refractory period. And yeah, there is a word for that, which is it is unidirectional. Just means one direction. Obviously, uni, one, directional, direction. Bam. Okay, so what are you guys saying? The threshold values behind cannot be reached because it's undergoing hyperpolarization. Lovely. How would you phrase that in a mark-worthy sentence? Good question. So I would say it cannot go backwards because the membrane is hyperpolarized, which means that a greater quantity of sodium ions would be required, or a greater concentration would be required to reach a threshold level, which is less likely to happen, and therefore your action potential will only go in the one direction, because if it goes the other way, it is more likely to have an action potential forming. I'm glad these are helpful, guys. So a neuron can't be depolarized more than once. It can be, but there needs to be a little time gap between, right? It can't be like instantly after. There needs to be a little gap. So not in quick succession, exactly. Right, how are you guys feeling with this? Are you feeling fairly confident? Is this making some sense? If you feel like you are totally getting this, send me either a nice little 1 or a nice little happy person. If you feel like this is like less okay, somewhere in the middle, it's not awful, it's not the best, you need to revise it, send me that, or a 2. If you feel like this little guy and everything's going wrong and you're wishing that you dropped your phone even harder, then you can send me 3. How are we looking? 3.9. Well, technically, yes, sir. That's not an option. So, okay, if you've not been taught it, yeah, okay, I understand that. I understand. I like that some of you are sending me more than 1. So you're being like, oh, yeah, I'm going to send 2 threes. Because it's like, you know, when you ask a kid, oh, do you want to go to the zoo? And they're like, yeah. And they're like, they put 2 hands up because they're so excited. I see you guys doing that. I know what you're doing. Cool. Right, let's have a look at an exam question, then. Let's see how you fare. Right, and one of these questions is a bit weird, so bear with it. So figure 1 shows changes in membrane potential of a neuron during one action potential. So what happens in the membrane to cause the change in membrane potential at time B? So at time B, what is going on to cause a change in membrane potential? Sarah, you need to consolidate. That is such a teachery term to use. I like it. Chloe, I need more information from that. You are being a little vague. So I'm talking about just B. I don't care about A, just B. So what are we saying? Just B. I'm just talking about B. I'm not talking about A. Cool. So some of you are saying the literal opposite of each other, but I see now some of you are actually getting to the right stage. So you need to say that, and I don't mind actually for this if you say voltage gated or not, because my mark scheme doesn't want it. But what happens to the sodium ion channels and what happens to the potassium ones? I want everyone to agree. Or I want to see that lots more people are saying the right thing than the wrong thing. So look, I'm up here, right? So think about my cell. Here is my neuron. Suddenly, it's becoming more negative inside. If it's becoming more negative inside, what's going out and what possibly is coming in or not coming in? Right, enough of you saying it. So sodium ion channels close. Potassium channels open, right? If the potassium channels are opening, potassium is going to move out, which makes it more negative. You guys know that sodium was moving in. Depolarization is when sodium moves in. If positive goes in, there is no way that it can be going downwards. Right? It'd have to be going upwards. Okay. If you want to say this in a slightly different way, as opposed to saying channels open or close, you can use the word permeability as well. So you could get marks on this question if you said the membrane becomes more permeable to potassium and it becomes less permeable to sodium. Okay, so this means sodium stops coming in. Yeah, potassium channels open, K plus moves out. Okay, cool. Right, this question is a bit of a weird one. So this is one you got to bear with. This is aimed mostly at Edexcel B, which is a bit weird of me because it's probably the one that the fewest people do. But no further action potentials can be produced between times A and C. What is the name given to the point between A and C? Yes. So I'm going to accept refractory period. And I was literally happy for just one person to say that before it all goes into chaos and anarchy when everyone's like, no, that's not the refractory period, because that bit's a refractory period. Yes, you are right that between A and C, this is the absolute refractory period for those of you who need to know that word, but everyone else can call it the refractory period. If I could change this, I would change it so I was only talking about this section here, but because I didn't have time, I did not. Okay, but well done for dealing with that. Have a round of applause. Patronizing much. So figure 2 shows the force generated by a muscle when it was stimulated by different frequencies of nerve impulses. So here's some nice information. A taser is a device used by the police to arrest violent suspects. It fires electrical impulses very similar to action potentials into a suspect. The frequency of the impulses is between 15 to 20 per second. So what is the effect that a taser is going to have on the muscles? So if I'm firing one at 15 to 20, I don't know, frequencies, whatever they are, pulses, waves, whatever, what's going to happen? Good. Spastication. I've never seen someone use that word, but I guess you're probably right. You mean spasming. So the muscles are going to spasm. They will contract and they will relax very, very quickly, which will cause them to twitch and spasm, essentially. So I like it. So it causes contraction. So that's literally a mark saying it causes contraction and relaxation very quickly. Should I write spasming? I don't know how to spell spasming. I'm not going to write spasming. I'm going to say twitching. There you go. So they are your marks. If you say contraction and relaxation very quickly, that is your sort of system of marks. Spasms. Maybe spasming isn't a word. Yeah, I like that. Spasms. Cool. Look, you can see here, this is worrying. So the force generated by a muscle, if you fire a taser at someone at 80 waves per second, they don't actually have this constant contraction and relaxation. This is just constant contraction. So if you fire a taser at someone, which is at a frequency of 80 per second, you will just kill them because you will stop you'll stop them from being able to breathe, because to breathe, you need your diaphragm, you need your external intercostal muscles and your internal intercostal muscles. And if it's all contracting, you can't do anything and you will die. So someone must have found this out quite a horrible way, I would imagine. Excellent. Right. I'm not seeing what any of these comments are, but part 2 then. So if we have a look at this, and this will be fairly quick, I want to start off because this is something that people get very, very, very confused about in terms of just what the names are of the different parts. And the different specs all use different terms confusingly as well. So out of interest, what could some of these different letters be standing for, which I've written on here to describe the different parts of my synapse? So don't even have a neurotransmitter. There's not even an N. How can I have that? Good. So there are vesicles. And let's say that they contain neurotransmitter. Double N? Probably. Oh, well. Okay, so vesicles contain a neurotransmitter. That is what that is. What else are you guys saying? Presynaptic neuron. Good. So presynaptic neuron. And there is also a postsynaptic neuron. Right, so the reason I've said this is because different specs have different things. So you might be used to calling this the presynaptic neuron, which is fine. You might be used to calling it the bulb, the presynaptic bulb. If you do OCR, that's what they call it. Although presynaptic neuron will be fine as well. Some call it the knob, which is incredible. I can't remember which spec that is, but the presynaptic knob, you can call it. And some of it just call it a synaptic membrane. Okay, so find out what your spec calls it and just use that from now on. Okay, most people will refer to this one as the postsynaptic membrane, not the postsynaptic knob, which is sad. What could my C word be? Careful here. What could the C be? AQA use knob too. Cleft, yeah, so you could call this the cleft. That's not actually what I'm going for. So this is called the cleft. Good. So there are calcium ion channels. These are also voltage gated. So well done for saying that, those of you that did. Voltage gated calcium channels right there. What could my S be? S is on the other side. I feel like I'm going to stop talking. Synapse, yeah, could do. The whole thing is the synapse. Well, the gap is the synapse, I guess. Not synaptic cleft. Something over on here. Good. Sodium ion channel. I'm going to, I'll write it out. There you go. Sodium ion channel. That is something else. Good. Lots of you were telling me, but this is acetylcholine. What is the name of this enzyme? That one. Acetylcholine esterase. Lovely. So commonly called acetylcholine. Yeah, I can't remember. Acid, ACHE. Thank you. Right. So, or ache. Not very too much like that. That is a fairly standard sort of abbreviation, similar to like NAD and FAD and NADH. I would go for acetylcholine esterase first. Finally, R. All I'm meaning by R is I'm going for receptors. And some of you will know about muscarinic ones. Some of you will know about nicotinic ones. I'm not going to talk about them for now. So that's for different parts. They're the different parts that we've got. In terms of how it actually works, what happens? So what's the first step which is going to happen to allow for my impulse to get to the other end? So before calcium gets there, there's no receptor up here. There's no, like stimulus that's coming. Something will come, but it's not a stimulus. Good. A depolarization or an action potential will arrive. Okay, so an AP will arrive. Action potential arrives. And then we can say that calcium, which is 2+, channels open. Okay. Essentially, that means that calcium will move in. So calcium is going to start to flood in. What does that cause? So channels open, calcium moves in. What happens next? Good. Vesicles, these little guys here, they will start to move and fuse to the membrane, to the presynaptic membrane, and release acetylcholine. Okay, they'll use exocytosis to do this. So vesicles containing neurotransmitter move and fuse, and they exocytose the neurotransmitter. Okay, so that's going to start to basically move in. What happens next? So my neurotransmitter, my acetylcholine, has got to this stage here. Where's it going next? Good, it will diffuse. So neurotransmitter diffuses across the cleft. Okay, so it will diffuse across the cleft. Okay, when it gets to the other side, it will bind to a receptor. And then, because this is so pedantic, and I'm sure you guys will know this, you want to say it binds to a specific complementary receptor. Yeah, same thing, same like terms you always have to use. Okay, what does that cause? So it binds. What happens next? Cool, yeah, lovely. So this will cause sodium to move in, causing a generator and hopefully an action potential. So sodium channels open, sodium ions in, generator potential. Hopefully it'll be enough to reach a threshold. Hopefully that will make an action potential. Okay, what happens next? From where do sodium ions come in? They're just going to be outside of the cells. There's sodium everywhere, all over your body. Good. So neurotransmitter is broken down by acetylcholine esterase. What is it broken down into? So acetylcholine hydrolysis, because we don't use the word breakdown in A-level biology, we use the word hydrolysis. It's broken down into what? Acetate or acetyl, I'll accept, and choline. So acetate or ethanoic acid. Okay, so it's broken down into acetate and choline, if you want to call it that. What happens to it now? So aceto, so definitely be careful, separate acetyl and choline. What happens now? So it goes back. Yeah, lovely. So it's going to be moved back into my presynaptic membrane. So moves back to presynaptic membrane, which will be endocytosis. And then final point, which lots of people don't know, is ATP is used to resynthesize acetylcholine. Okay, so in terms of what everyone knows, most people are good with the first 5, maybe the first 4. Everyone always forgets this, and everyone always forgets this. So there are 2 more marks if you ever have an exam question on this. Out of interest, what is an inhibitory postsynaptic membrane and potential? What are these things all about? Both of them are taken back, and then they are remade into acetylcholine using ATP. Okay, then they're stored in a vesicle forevermore. So inhibitory postsynaptic potentials, what do these do? What's different about them? So it stops an action potential, lovely. So essentially, some neurotransmitters, when they bind, they don't cause sodium to move in. Some of them cause chloride ions to move in, which basically hyperpolarizes it. It means it's less likely for an action potential to occur. So we can say something along the lines of these make it less likely for a action potential to occur. And that's how your eyes work. Okay, and a good example of that is a neurotransmitter called GABA. And the one in your eyes for EdXL people will know about, it's called glutamate, which can actually be excitatory as well. But anyway, right. How are we feeling, guys? Yeah, GABA is the example, spot on. Snow White and Emily Winter, I feel like you two belong together with your names. Oh, look at all those 1s. If I just stop looking now, then everyone's been a 1 so far. Snow White, 1, 1, 1, 1, 1 is actually quite a high number, so not quite ideal. 2, Abrish, why? Missy, no. Lucy, no. Right, I'm just going to stop looking. Most of this was 1, so if I stop looking, that makes everything better again, right? Okay, final question then. Describe how calcium ions are involved in synaptic transmission. Charlie, you are clearly hilarious. Well done. I think of another number you could put, which would be even funnier. So calcium, yeah, lovely. Calcium will do what? So it causes vesicles to move. What's it do first do? Causing vesicles containing neurotransmitter to fuse. But before, it needs to do what? It needs to move in, lovely. So calcium moves in through channels. Voltage gated channels. There you go. Good answer. So it moves in through voltage gated channels, and then it causes it causes vesicles to basically move, fuse, and release acetylcholine. Okay. Cocaine changes the way that some synapses function. Figure 1 shows a synapse in part of the brain. The synapse uses a neurotransmitter called dopamine. Bear with me. Okay, this synapse only transmits information from neuron A to neuron B and not from B to A. Give one reason why. Why is it the case that a synapse can allow an action potential to only go in one direction? Cocaine used to be prescribed as an eye medicine back in the Victorian times, for anyone who cares. Good. Lucy, you are spot on. The reason that a synapse can only cause an action potential to go in one way is because the only receptors are on one side. They're only on the postsynaptic membrane. Okay, so receptors are only on the postsynaptic membrane. Okay, that's a really good answer. Refractory period, feasibly, does that get a mark? No. So refractory period won't give you a mark here because by the time all these vesicles have diffused, it's probably reset itself. So it's more that the receptors are just on one side. And you could also say that the vesicles are just on one side too. Because this side doesn't have any vesicles, you can't get any movement in that other direction. And because there aren't receptors, it can't kind of turn itself on that way. Explain why cocaine is able to bind to the dopamine transporter as shown in figure 1. So why is it that, so I think, hang on, here is cocaine, this like shape thing here, and it can bind to the transporter thing here. Why can it bind? Good, they have similar shapes, so can fit together. So similar shapes for one mark. So they can fit together for the other. Bam, lovely. So specific complementary, complementary I would accept. Specific, yeah, I think that's probably fine too. So dopamine is released at synapses in parts of the brain where pleasure is perceived. Using information from figures 1 and 2, explain how the use of cocaine can result in feelings of pleasure. Look at this diagram before you tell me anything that's silly. So look at this diagram. What is cocaine doing? Why is that going to make you feel pleasure? What does the dopamine transporter actually do? So think about what the dopamine transporter does. Look at where it is. Look at what it does. What is cocaine preventing from happening? Holly, you are spot on. Well done. It doesn't bind to a receptor. Look, it's not binding to the receptor over here. Yes, it's not binding over here. It's binding here. Look. So what's that stopping from happening? No, dopamine isn't released from these receptors. Dopamine's released from vesicles, isn't it? But what do those receptory thing, not receptors, what do the transporters do? They move the dopamine back in, right? So cocaine works by preventing you from taking the dopamine back in. It can be released, but it can't get back in. So dopamine is going to stay bound to your receptors. It's not going to move back in. So you're going to get this sustained feeling of pleasure for a really long time. Well, not a really long time with cocaine. So cocaine blocks transport of dopamine out. Dopamine transport out. Okay, what's that going to cause to happen to the dopamine concentration? What's going to happen to the dopamine concentration? How do I know? I'm a scientist. Terrible minds all of you have. I'm a scientist. I'm allowed to know things about drugs. Disgusting. Good. So if you can't take it out, you need to say for your second mark, cocaine concentration increases. So it's going to build up really, really slowly. And if the dopamine is there, so when we were saying cocaine, I meant to say clearly something's in my mind. Doesn't help my cause, does it? Dopamine concentration will go up. If my dopamine concentration is up, what's going to happen on this side? So it's not moving back in. If it's really high up here, what's it going to do? So it's going to stay high in the cleft. It's going to do what? No, Lucy, no. I'll go and explain again. So cocaine is this sort of black triangle shape thing here, right? Or this T-shaped thing. It binds to this transporter. So dopamine cannot move back in. So the concentration increases. What does dopamine do when it's in here? Good, it's Olivia, thank you. So this means dopamine will bind to the receptors. Yeah, so bind to the receptors, probably say add the caveat for longer or something. Yeah, so that's why cocaine has this effect. That's why loads of drugs have the effect. They block your ability to take it back up again. So the concentration gets high, stays bound to receptors, and you feel whatever is associated with that neurotransmitter. In dopamine's case, pleasure. Okay, cool. Right, so something that I need to tell you about very quickly. So you should now know what a resting potential is, should know what an action potential is, and you should know how synapses work. But to do some plug-in to make it worth my while to have sat here lovingly and told you about cocaine and various other things, you guys should be aware that we are doing some seminars in London in April. So if any of you are interested in hearing me talk a little bit longer than just your 1 hour a week, which is what you get, I'm doing an exam crash course, which will basically cover a massive proportion of the A level, and conveniently it's just before your exams. So if you're in year 12, I'm going to be covering the role of proteins, cell division, mass transport in plants, which people tend to hate, mass transport in animals, exam technique. You get a break time. You can go and wander around London for a bit and get some food for 15 minutes. Or for this one, another 15 minutes. Oh, sorry, there's a lunch break there. There you go. See? Treating you. 45 minutes to go and get some food in London. Also, there is one which is for year 13s, which is similar. So they're both in the Imperial College Lecture of Theatres in the Huxley building. This one's going to be on calculations, photosynthesis, inheritance, DNA tech. And before anyone says, oh my God, it's £90. It's not, it's £99. But also, you guys will very soon be finding out that tuition costs an awful lot of money when you go to university. So I remember being told in my first ever lecture that the price per lecture is £50 based on 9 grand divided by however many lectures you have. And that basically freaks me out into pretty much going to every lecture. So it is expensive, and I do understand that. However, education tends to be quite expensive. You guys are just very lucky that you go to schools which are paid for by taxes and stuff, whereas this is not. Your parents' taxes does not pay my wage, unfortunately, as much as I wish it could. Anyway, for those of you that are interested, you guys should have a look at this. We are also doing sessions on chemistry. In fact, let's look at all of them. So that's me. That's meant to be my face. Georgia, who you guys might know, who does chemistry, she's going to be doing some. Max, who does maths, is going to be doing some. And Matt, who does physics, is also going to be doing some. And I'm pretty sure there is some discount code if you go to more than one of them. So if you want to go to all of year 12 and 13, biology and chemistry, you will be able to get a discount code if you email us. So our email address is, well, I'm sure my person who is writing comments for me, Snap provides a person writing things. There you go. He's given you a email address if you guys want a discount. If you live in Dubai, it's going to be very expensive for you to get here, but I'm looking forward to seeing you. Oh, are you doing these videos every week? Yes, so these other videos I am doing every week. So once a week on Wednesday. And if you want to sign up to Snap Provise, I teach, how many lessons do I do? 1, 2, 3. I do 4 a week. That's it. Do you need to know about cocaine? You do not need to know about cocaine, Aisha. It wasn't a apply question. What's the next video? I made one today about genetics, because people don't like genetics. I'll do DNA tech, not just yet, because not everyone's done it yet, and I don't think it will help everyone, but I will do it, don't you worry. Right, anyway, I am going to go, I think. So have a lovely evening, guys, and hopefully I'll see you at some point in the near future. Adios.